So my name is Julia Maxson, and I'm an assistant professor in the Knight Cancer Institute at Oregon Health and Science University. And the title of my project is Mechanisms of Kinase Inhibitor Resistance in Chronic Neutrophilic Leukemia. In order to develop new targeted therapies, you need to know what the targets are in the cancer. And so when I was a postdoctoral fellow, I actually discovered that there was a rare kind of leukemia called chronic neutrophilic leukemia, in which almost all of the patients have mutations in one particular gene. And this gene, CSF3R, basically receives the signal that tells you that you need to make more of a particular type of white blood cells called neutrophils in order to fight infections. And so, when you need more of this white blood cell, the gene turns on, sends a signal, you make more cells. And then when you don't need more cells, it turns off. But in this disease, almost all of these patients have a mutation that causes this particular gene to be stuck on all the time, almost like if you broke the light switch on, right? And so the result of that is that these patients' blood systems just keep producing more and more and more and more of this white blood cell. And so that's a problem because it becomes a cancer. And so we found that these patients have this light switch on, but we also found a drug that can stop that signal and turn it back off, even though it's sort of broken. And so that was really exciting. And fortunately, this drug was already FDA approved for another kind of cancer. And so my sort of fantastic clinical collaborators here at OHSU and around the country were able to run a clinical trial for these patients with this rare leukemia. And what they found was that this drug actually causes the leukemia to respond in about half of cases. And so, that was, really exciting because we found that the patients that have this mutation have about 50% chance of responding, and the patients without these mutations, by and large, don't respond. So that allowed us to really understand, that this targeted therapy was paired with these mutations. But some of the patients don't respond. And sometimes when the cancer responds, it comes back later. And so the goal of this proposal for the foundation is to really understand, why do some patients respond and others not? And then why do some patients initially respond and then have a relapse? And so we found some mutations that seem to go along with relapse, and mutations change the disease to make it harder to treat. So this is actually a really rare form of leukemia, so it doesn't impact a ton of patients. But in those patients that it does impact, it's really deadly. So best estimates would say that there's really only like a 2-year survival after diagnosis for this. And I know it's a real rare leukemia, and so you might ask, why would you study this rare leukemia and not a more common leukemia? I guess for me, I feel like, we've made really important strides in understanding, what goes wrong in this cancer and how might we reverse it. And so I kind of feel like a personal commitment to this particular group of patients, and really, because it's something I've worked on for a long time, really, wanting to take it to that next level and provide therapies that don't just work for a window of time, but might be curative or might, provide really long-term remissions for patients. So even though it's a rare disease, for me, I feel like a personal commitment to helping those patients with that cancer. So mutations in CSF3R are highly enriched in chronic neutrophilic leukemia. They also occur more rarely in a related disorder called atypical CML. And then also we find that mutations in CSF3R occur in a type of pediatric leukemia called pediatric AML. So there are other projects in my lab that work on the sort of AML side of the story, but that is also something that we're really interested in enhanced therapeutic development for, because what our clinical collaborators have found is that in AML, CSF3R mutations can take a subset of patients that would normally have favorable prognosis and turn them into a subset of patients that have unfavorable prognosis. So even though it's not as frequently mutated in that disease, it's very important in terms of prognosis, and those patients have real clinical need for better therapy. And then mutations in SETBP1 are found in a lot of different kinds of leukemias. So they occur in about half of patients with chronic neutrophilic leukemia and approximately a third of patients with atypical CML. But they also occur across the spectrum of myeloid leukemias. So they tend to be enriched in myelomonocytic leukemias as well. So chronic myelomonocytic leukemia and juvenile myelomonocytic leukemia. So what we're really hoping here is that we can understand, what causes drug resistance so that in the future, we might be able to develop a therapy in which resistance doesn't occur, or we might be able to use more than one drug in combination to prevent the development of resistance. So drug resistance is a problem across leukemia, right? And so here with chronic neutrophilic leukemia, we have a really defined context where we know who the mutations are and who the players are. And, that gives us like a really controlled system in which to study how drug resistance occurs. And so our hope is that, if we can get a handle on it in this context, that those lessons might be applicable in other types of blood cancers. And then our hope is that in the second year, we'll continue that work and use those models to both understand biology, but also start to get a sense for how that contributes to drug resistance, and then maybe hopefully in the future how we might be able to reverse that to resensitize the patients to drugs or to be able to kill the cancer cells more effectively so it doesn't come back.